So good morning, everybody. It's my pleasure to welcome you on this session on the room number 2. And the session will be about metaomics and microbial genomics. So we will have the pleasure to listen to three, in my opinion, very interesting talks to come. First, I would like to present Clémence for you. So Clémence, she's from Bordeaux. From INRIA Bordeaux, and she will present a work that was published last year or this year, I forgot, in the Journal of eLife, which is about the metabolic complementarity between microbes in the way of understanding how these microbes shaped community. So, Clémence, the floor is yours.
Good morning, everyone. I would like first to thank the organizing committee of JOBIM for the opportunity to talk. I will present methods dedicated to assess the metabolic complementarity and identify key players in microbiomes. Microbiotas are surrounding us in our body, but also in the ocean or in the soil. They consist of thousands of species that interact in a shared environment. These species, among which many have never been cultivated, form a complex network driven by these interactions occurring between members. Interactions are largely mediated by metabolites that are both produced and consumed by all of these species. Therefore, the diversity of functions in a microbiome is a key to understand how these ecosystems live or react to perturbations. Most microbes cannot be isolated, so omics experiments are the main source of information. In the past, we mainly wanted to answer the question who is there using amplicon sequencing, but now shotgun metagenomics becomes routine, enabling us to get all sequence from a sample and answer the question what might microbes do in this particular ecosystem. We can reconstruct genomes from metagenomes that are called MAGs. And with that, it gets possible to apply similar methods to those applied to individual genomes. One of them is the reconstruction of metabolic networks that I will discuss today. A metabolic network is a set of metabolic reactions, like this one here, transforming a substrate A into a product B. Metabolic reactions are catalyzed by enzymes, here. And at genome scale, a metabolic network actually contains all reactions associated to any enzyme found in the genomic sequences. Usually, that makes a set of 1,000 to 5,000 reactions. All these reactions are the functional repertoire of the cell. What could an organism do? We are in a context of communities of organisms living in a shared environment. Let's assume we have a genome or a MAG for each of them, so each of our two bacteria. What we would like to do is build a metabolic network at genome scale for each bacterium. Using these metabolic networks, we could identify complementarity between the metabolism of each species and suggest possible cross-feeding interactions. Here, the green bacterium produces D, the blue one consumes D. This could be a cross-feeding interaction. And in case we are interested in a given function, here G, we would like to find key species, key players that are associated to that function. Here, clearly the blue bacterium, but also maybe the green one because it can produce D that is needed by the blue one. So we can't find these interactions just by looking at the metabolic networks. They are static objects, and if we want to simulate metabolic behaviors in specific conditions, specific environments, we need to apply mathematical frameworks to our graphs. Here, we chose a qualitative modeling called network expansion that recursively computes reachable or producible metabolites starting from a set of available nutrients. Here, A and B being nutrients, C, D, and E are the set of reachable or producible metabolites. We implemented this algorithm in answer set programming, ensuring scalability to a large number of metabolic networks, and we used it in our pipeline that I will now present to you. This pipeline is called MetaJ to Metabo, or M2M, and I will show you its main steps. Starting from a collection of annotated genomes, or MAGs, M2M automatically reconstructs metabolic networks for each of them. We use pathway tools and the Metacyc database to do that, and we actually built a Python wrapper to ease the automation. Our first analysis then consists in establishing and comparing the individual metabolic potentials of each microbe in its environment. But we know that organisms actually interact with each other. So we compute the collective metabolic capabilities that take into account the complementarities between metabolic networks. This is actually putative cross-feeding interactions that we take into account at that step. That way, we can identify metabolites that are producible only through mutualistic interactions and can therefore not be produced by a single microbe. Here are the green and yellow stars. These metabolites, or any others, can be set as metabolic targets for minimal community selection. The objective is to determine communities that can produce compounds of interest, so our green star or our yellow stars. Altogether, this pipeline, M2M, aims at systematically screen the metabolic potential of microbes and the mutualistic potential of a large community. The main step of our pipeline is the selection of minimal communities. This is a combinatorial problem solving, reducing the complexity of an initial community of species, living in an environment where nutrients are available and with a metabolic objective that is producing a set of compounds. So we can find a minimal community, but it's very likely that one minimal community will not be the only solution to the problem. Much more likelier, we will have a lot of equivalent minimal communities, up to millions, because the metabolism is highly redundant. So we have a huge combinatorics to deal with, and enumerating all minimal communities as well as analyzing them is very demanding. So instead, we propose the concept of key species that can be very easily computed and is the set of species found in at least one of the predicted minimal communities. So here we have 4 optimal minimal communities, but altogether only 5 symbionts occur in them. And we can further distinguish key species into 2 categories: essential symbionts that occur in every minimal community, like the yellow symbiont, or alternative symbionts occurring in some but not all communities. As it is very efficient to compute, we believe that the key species concept addresses the combinatorial challenge brought by the functional redundancy observed in natural communities. We applied our pipeline to a collection of 1,500 reference genomes from cultivable species of the human gut microbiota. We built metabolic networks for each of them and looked at metabolites that could only be producible through cross-feeding. We classified these 156 metabolites into 6 categories: lipids, sugar derivatives, etc. And we made them metabolic objectives for minimal community selection. For each group, we computed the key species among the initial 1,500 species, but we also enumerated all equivalent minimal communities. to evaluate the combinatorics of the problem. So here we have our 6 categories. The minimal size of a community is ranging from 4 to 11 bacteria. And you can see that we have up to 7 million equivalent minimal communities for sugar derivatives. Fortunately, the number of key species is way smaller. For example, 185 species out of the 1,500 for lipids metabolites. And the number of essential symbionts is even smaller. We studied how these key species interact and connect with each other in communities by studying the composition of every enumerated communities. It's very hard to represent 58,000 of 7 million equivalent communities. So we built association graphs and compressed them using power graph analysis. So that way we compressed clicks and bclicks motifs in network, ending up with representations like the one you have here. So small nodes are species, key species, and large nodes denote groups of species with an equivalent role in the communities. They were compressed during power graph computation. For the example of lipids, we have 58,000 communities, and each of them is composed of one species from the node 1, one species from the node 2, this species from node 3, which is an essential symbiont, one species from node 4, and the 3 species from node 5, which are also essential symbionts. In addition, when we looked at the taxonomy of the species, we observed that groups of equivalent bacteria in large nodes here usually share the phylum, suggesting that they carry functions that only specific members of the phylum also carry. So here, for example, lipids production needs a function carried by this group of Bacteroidetes. This is a reading pattern that we observe for all groups. And when we went back to biology, we observed that essential symbionts were sometimes shared between several groups of metabolites. And we found that some essential symbionts are actually already known and studied in the context of probiotics. So the first experiment I showed you was performed on reference genomes, but MAGs are the main genomes available for microbiomes, and we know that they are possibly incomplete or contaminated. So we designed an experiment to assess the impact of MAG's incompleteness on metabolic network reconstruction and subsequent modeling. To do so, we removed between 2 and 10% of genes in all or subset of our initial MAGs. Then we ran our pipeline. Regarding metabolic network reconstruction, so we observed, as expected, that the number of genes in MAGs is lower than the average number of genes in reference genomes. But this cannot be seen as after metabolic network reconstruction. They all have similar numbers of reactions, pathways, and metabolites. So that's good news. What happens when we model the communities? There is a strong metabolic redundancy in microbiotas, and this reflects on a very stable set of compounds producible by communities, regardless of the degradation of the genomes. Some effects are more visible when we look at key species computation. There, the more we degrade the genomes, the more different are the sets of key species. But we can see that for small degradations, the set of key species is relatively stable. So we can conclude that the criteria typically used for MAG quality, which is more than 80% completeness, is likely to be sufficient to get a coarse-grained yet valuable and reliable first picture of the metabolism in a community. As a summary, MetaJ2Metabo is a flexible pipeline for large community analysis at the metabolic scale. is very flexible. You can reconstruct metabolic networks in the pipeline or provide them as input. Each step can be run individually. The purpose of the pipeline is to get a first picture of the metabolic complementarity and metabolic redundancy in a large microbiota. Its second objective is to reduce the complexity of the initial microbiota by pinpointing members of interest for targeted functions. In my presentation, I illustrated the use of M2M for genome or MAGS collections. However, another use case that is presented in the paper is to build a community by sample in a metagenomic cohort, one sample, one community, and then compare them. So we did it for samples in the MetaHit dataset. Finally, just to remember that we look at might be producible, and we would need metatranscriptomics to further refine predictions in the context of each environmental condition. With that, I would like to thank you for your attention, thank all my colleagues and collaborators, and let you know that we are hiring postdocs in RIA Bordeaux. Thank you.
Thank you very much, Clémence. Don't see any. So again, if there is any question, don't hesitate to ask them in the chat. But before them, I could start with. I have plenty of them, but I will start just maybe with the previous one. You were showing a very interesting result to me about the especially the MAGS and the fact that you could use the M2M pipeline to better understand the quality of the MAGS and something like that. Could you have any? Could you give us any idea about? In my opinion, it could be a bias because of the metabolite that you are looking, because some of them, they are associated to pathways that are very well known versus others that are less known. And could you say a little bit about if to you, you could use this pipeline as well to, let's say that for some pathways that are very well known, I think your approach is very cool, but also to emphasize the pathway that need to work on, because I don't know, or if in your RD there is a ranking of the pathway that you could trust versus the one that you cannot trust at all, and especially you were talking about different kind of metabolites, and maybe using your pipe, you see maybe different kind of results. I don't know if you could give us some idea about what is behind the scene at some point.
Okay, so on that topic, we highly depend on the initial database that was used for metabolic reconstruction. So the wider the database, and by wider I mean containing a lot of reactions, even those that belong to secondary metabolism or very poorly known functions, the better the metabolic network reconstructions and the predictions. So to precisely answer your question, I'm not sure we can really look at the pathways that are better reconstructed because they are better known and compare them to the pathways that are less known. But we could definitely look at the pathway composition of the metabolic networks and see for each species whether it's more complete or less complete when we degrade the genomes. So that we could do. But then we really rely on metabolic network databases to get poorly known functions.
And so I take my shot for another question because I don't see any. Sorry, I'm not necessarily used to that very much, but anyway. And about the number of solutions that you have, do you have? Maybe I missed something during your talk, but do you have the same complexity of solutions based on the metabolite that you were looking at? You were comparing the lipids, the carbohydrates, and so the compression graph that you were showing. Do you have a different complexity based on the metabolites of interest?
So no, because we ranged from, I think, 1,000 solutions for one particular group of metabolites to 7 million of solutions for another group. So there are several explanations, possible explanations. The first one is, of course, the number of metabolites that you put in your metabolic objectives. And then whether these metabolites occur in very rare pathways that are present in very few members of the community, or in pathways that are, I don't know, maybe widely present in all bacteroidetes, for example, then you increase your combinatorics. So it's not necessarily related to the type of compounds you look at, but rather really the structures of your metabolic networks.
Okay, thank you. Okay, so I think we have to move on, but again, if people are too shy, don't hesitate to type your question on the chat. Clémence is still there, you can answer the question in private. Sure. A question from Samuel Chaffron. So I don't know if Clémence, you could see the question. So you talk about the objective function for identifying minimal communities. Could you give more details about what are these functions and how you select them or how you build them?
Yes. So maybe some of you are familiar, and Samuel is certainly is, with objective functions in flux balance analysis. So in our case, objective functions are way simpler because we are using qualitative methods. And an objective function is simply a set of metabolites that we are interested in producing. So in the examples that I showed, it was, for example, a set of carbohydrates. So we just put these carbohydrates identifiers, and then the community selection will be performed to ensure that these carbohydrates are producible in the reduced communities.
Okay, and we take the question of Roman as well. Is it possible to integrate the temporal aspect to M2M? Because ecosystems, I mean, there is a temporality at some point, and so could you introduce that in your pipeline?
So far, not really, but in a certain way, because if you are looking at longitudinal studies for cohorts of individuals, you can in some way take into account temporality by comparing the metabolites and the communities at each step, but it's not taking into account temporality otherwise.
Okay, thank you again. So we have to move on. It was very interesting. Thank you again, Clémence. Again, if there is more questions, don't hesitate to use the chat, and Clémence could answer there is another one left in the chat, Clémence, if you could answer it. Excellent. So the next speaker is Benoît. So Benoît is from Maillage in Paris-Saclay. He will discuss about the comparison between different sequencing, metagenomic sequencing. So to compare the shallow depth metagenomics compared to the more standard whole genome sequencing. So Benoît, the floor is yours.
Hi everyone, I'm Benoît Goutor, and I will talk to you about shallow shotgun metagenomic as a cost-effective and accurate alternative to whole genome sequencing for taxonomic profiling in a clinical context. First, a few words about human microbiome. It consists in all microorganisms colonizing our body. I'm focusing on bacteria, but there is as well archaea, fungi and viruses, and they form a symbiosis interaction with ourselves. But when things go wrong, it has been proven to have some links with several diseases, such as metabolic and autoimmune diseases, bowel inflammations, cancer, or more recently in neuropsychiatric disorders. It can be very useful in a clinical context as a non-invasive diagnosis or prognosis tool, and as well because we can act on it through interventions like diet interventions, probiotics, or fecal transplantations. To study this ecosystem, we have several sequencing strategies. First, metabarcoding consists in PCR amplification and sequencing of a phylogenetic marker gene. we often use 16S for bacteria. It's useful to characterize intra and inter-sample biodiversity. We can build taxonomic profiles and it's very cost efficient, but it has some limited taxonomic resolution, meaning that we cannot always identify the species, but only the genus or the family. It has some limited taxonomic coverage, meaning that some populations may be lost, either because they don't have the gene, either because the primers we used for the PCR did not match with their sequence, and also it has some amplification and gene copy number biases. On the other hand, shotgun metagenomic consists in sequencing all the DNA we find in an ecosystem. We can reach a way better resolution of taxonomic profiles. We can identify the species or even the subspecies. We can build functional profiles by identifying genes and pathways, and we can also reconstruct de novo genomes called metagenome-based assembled genomes or MAGs, but it's very costly in terms of sequencing and data processing. There is as well other analysis techniques, but I will not go into detail now. In clinical research now, we have to face a huge variability between patients, and we have as well new insight into the microbiome dynamics and plasticity. So we need to include many patients in our study or to have a longitudinal approach, meaning that we will need several time points for the same patients, and as a consequence we have many samples to be processed, and metabarcoding is often the only choice to carry out large-scale studies. To give you an idea, here is the number of hits we find in PubMed for human gut microbiota and shotgun sequencing in red or 16S in green, and we can see that 16S is still widely predominant even today. So there is a need for alternative techniques. Shallow shotgun metagenomic is one of the techniques that could fill this gap. It's just like shotgun sequencing but at very low sequencing depths. Typically less than 1 million reads per sample are needed, while in deep shotgun sequencing we will require 20 to 40 million reads per sample to study a human gut. As a consequence, we cannot perform any assembly of the read and we'll have to use a mapping-based approach. Compared to 16S, we can reach a fine resolution taxonomic profile. We can build functional profiles and we don't have the biases related to the PCR. And compared to deep shotgun, it is cost efficient, but there is a need for a representative catalog to perform the mapping, and some rare populations may be lost due to low sequencing depths. Here is a paper published by Hillman and his colleague in 2018. We can see that they could reconstruct nearly the same information at 50 million reads per sample and 500,000 reads per sample. The color refers to the body size. And for stool samples, they could reconstruct the Shannon diversity with as few as 10,000 reads per sample. There are still some open questions, and we wonder whether shallow shotgun metagenomic was suitable for clinical studies. To answer that, we first used simulation to see how we could optimize the precision and the reliability of taxonomic profiles at low sequencing depths, and then we would use the real data sets to see what information could be recovered at the sample level and at the cohort level. This is the pipeline we used. As an input, we have either simulations or real data sets. Then after a pre-processing step, we use subsampling to mimic low-depth sequencing. Then we do a mapping on a catalog called Unified Human Gut Genomes. This catalog was recently published and it gathered data from metagenomic studies, culturomic studies and public databases, and they gathered over 200,000 genomes that they clustered into 4,600 species. These species, they figured out that most of them didn't have any cultured representative. They are shown in green in this figure. So it's very important for us because thanks to this we are able to identify an uncultured organism. So after the mapping we will figure out that we have to perform a filtering on the hits, and we will discuss further about that. Then there is a step of reallocation of ambiguous reads. I will not have time to talk much about this, but if you have questions, I will be happy to take them. And finally, we obtain our taxonomic profiles. For the simulations, we retrieved 100 taxonomic profiles from curated metagenomic data. We had an average diversity of 112 species, and we simulated 10 million reads per sample. When we perform the mapping, we realize that we have reads that map to several genomes. We have some genomes that are really well covered, others not. So to label the reads, we first see how many hits do they have. If it's 0, we label them as unmapped. If it's only 1 hit, we label him as unambiguous, and more than 1 hit, ambiguous. And then we look if the target is in the hits, and we can label this as a true positive or false positive. This is all the genomes hit by mapping. On the x-axis you find the fraction covered by unambiguous reads, and on the y-axis the unambiguous read counts. You can first notice that there is a huge amount of false positive shown in red that need to be filtered out. To do so, we could put a threshold on the read count or on the fraction covered, but we can notice that we don't take benefits of the shape of this scatter plot, and here we could retrieve some true positive that are relatively rare, so few read counts but well covered. And to take benefits of this shape, we used machine learning techniques. We used linear discriminant analysis, logistic regression, and random forest to perform a task of classification. As an input, they have the read count, the fraction covered, the specificity ratio, which is defined as a number of unambiguous reads mapped to a genome divided by all the reads mapped to this genome, and the sequencing depth. As an output, it says that if the hit corresponds to a truly present species or a spurious hit. We use the fourfold cross-validation process and automatic threshold syncing that tolerate 10% of false discovery rate in order to compare method and sequencing depth. This is the area under the curve of the classification task. Using first only basic features such as read count and fraction covered, we have a AUC of 85 and 90% respectively, and we see a true enhancement while using machine learning technique, and especially random forest based filters that reaches an AUC of 97%. Even though we have an efficient filtering, there are some false negatives, so species that are expected in the profiles but not found, and this is their distribution regarding the proportion in the expected profile. We can see that when we move to dark colors, so that we reduce the sequencing depth, we have more and more false negative, but they mainly concern rare species. So for instance, if at 500,000 reads per sample, we identify most species beyond 10 to the power of -3. Back to real data now. We wanted to measure the effect of sequencing depth at different levels, at sample level and cohort level, and to apply our random forest based filters to see how it behaves on real data, and to compare it with a basic filtering technique which was inspired by what was done on a previously published paper on shallow shotgun metagenomic. What we could see is that this filter performed really bad on the simulations, with a false negative rate of 40% and a false discovery rate of 45%. This is the datasets we used. We have 3 datasets covering different continents and clinical conditions, and every time we have 2 groups of patients, and we want to find differences between these 2 groups. In the first case, in Lumba, we have a patient with hepatic diseases, and we want to discriminate the one with advanced stage and early stage. In Matson, we have patients with cancer, and we want to predict whether they responded to immunotherapy or not. And finally, in Chin, We have patients that have liver cirrhosis versus healthy controls, and we have as well something interesting with a discovery and validation cohort. Here is what we obtain when we apply the random-based filters. This is at 10 million reads per sample. We can see that we can recognize the shape of the mapping hits. We produce profiles with an average sample richness around 100 species per sample, and we have a species relative abundance that goes down to 10 to the power of -4, so we can identify some rare species. Back to what interests us in the first place, I mean the impact of the sequencing depth on the results. Here is the diversity we can measure in each sample. On the x-axis it's what obtained at full depth, and on the y-axis what's obtained with subsampling at different levels. On the left part it's the richness, so the number of species that were identified. We can see that it's strongly impacted by the sequencing depth, so the fewer we have reads, the fewer we detect species. But on the right part it's the Shannon index, which takes into account the proportions of the species, and this is way less impacted by the sequencing depth, so meaning that the overall profile is quite well reconstructed. At cohort level now, so we try to see if there are differences between groups of patients. The first test we perform seeks for differences in terms of biodiversity, so we display the p-value of an ANOVA on Shannon indices regarding to the sequencing depth. The first thing we can see is that there is a strong effect of the dataset, of course. So for instance, in Matson there is no signal, while in Kin there is a strong signal. The effect of the filtering technique can vary across datasets. For instance, in Kin, the lowest p-values were obtained by random forest-based technique, while it was the opposite in Lumba. And what interests us in the main place is the effect of sequencing depth, and we can see that the p-values are relatively not impacted by the sequencing depth, down to 500 reads per sample. The second test tries to see if two patients of the same groups tend to look alike more than two randomly picked patients. So it's called a PERMANOVA, and it's based on Bray-Curtis distances. And we can see again the same kind of results, so everything depends on the data set, and the effect of the sequencing depth is quite negligible, down to 500 reads per sample. Finally, we tried to see if we could predict the groups of the patients according to their microbiota, so we performed the classification with random forest, and we display here the area under the curve according to the sequencing depth. For Matson we could not perform this classification because there were no signal, we saw that before, and for Kin we could split into the discovery cohort and the validation cohort. So again we can see that the effect of the filtering technique depends on the data sets, but the classification was possible and the AUC were not strongly impacted, down to 500 reads per sample. A few take-home messages to conclude. So first, machine learning based technique really enhanced the reliability and the precision of the taxonomic profiles. About shallow shotgun metagenomic, it was really suitable to study human gut microbiome, and it allowed to recover well the biological signal in the different data sets we studied, but some information is lost, mainly about rare species, if we want to control the reliability and the interpretability of the results. Some limitations about the filtering strategy. As it relies on the training step, it needs to be applied on the same conditions that were used for the training. These conditions are in terms of the diversity of the ecosystem we study, but as well on the distribution of the abundances and of the read length. And more generally about shallow shotgun metagenomic, as it relies on mapping-based approaches, it requires a really good representative catalog. It's not a problem in human gut microbiome, but it may be a problem in other applications. Finally, I would like to thank all my collaborators in Maillage, in Laboratoire Alphabio, and in CRCM, and Miguel and Disk Platforms for the computational resources, and Anerte and Alphabio for the funding. I would be happy to take any questions. Hi everyone, I'm Benoît Goutor.
Thank you, Benoît. So yeah, thank you. It was very interesting. So again, same processes. If you have any questions, don't hesitate to put them in the chat. But I will start before people were not too shy to put them. I have a question about the rare species. So I was very intrigued by the fact that so okay, you cannot catch the rare species using your approach. That was pretty obvious. But I was wondering if you were trying to put in this to dig more in this direction so is it always the same rates of rare species? So do you have any idea about the, let's say, the threshold of rarity that you could not catch, or is this something that is not reachable right now?
It really depends on the amount of false discovery rate you tolerate, because for instance what I could see like in literature or stuff, you can put some threshold where you will catch a lot of rare species and you will be really happy with the fact that the sequencing depth doesn't have any impact, but you will have a profile with a lot of false positives, so it may be a problem for a lot of a lot of downstream interpretations or stuff. And we with the strategy we used, we really wanted to master at each sequencing depth to control the false discovery rate, and so we had these thresholds that were applied and where we looked for thresholds automatically, and then we could control that, and then we can clearly see the impact and the rarity of the species we can reach according to the sequencing depths, and we can see that the false negative increase and increase. That's what we show on one of the slides.
Okay. Yeah. Okay. Yeah. That's cool. And so in following this question, so could you recommend with I don't know, maybe it's a stupid question also, but could you recommend, because there is this comparison between these different kind of sequencing. So do you think that there is like a decision process that you could go in advance to the biologists? Like what kind of sequencing do you recommend based on the disease, or I don't know, because you were working mainly in human microbiome where it's pretty homogeneous. And at the end, you were talking about that it's not necessarily tractable to put it in other kind of environment. But I would say, what is the reason? Do you need something more? It's because of the stability of the environment, or you think it's more about the what we understand about the environment? And so could you guide
the most practical stuff is that you need a very good reference catalog, and you need to trust it and to be to have to rely on it. So that's really like the first step and the first limitation regarding other applications that may concern in microbiome studies. And then it as well depends on what you're looking, as you will only identify the organisms that have the more DNA in your ecosystem. If you study like rare viruses or stuff, you need to forget about that. And as well, if you want to do a deep functional study that needs assembly of genes to look for SNPs or stuff like that, it's not suitable. But in clinical research, where sometimes you just want to have a first view on an ecosystem related to a disease, and to me, it seems very interesting in comparison with for instance, 16S or stuff like that.
Okay.
But what was really interesting is that the groups, the structure of the data sets were not really impacted by the sequencing depths, and so if you plan to do this kind of analysis for the same amount of budget, I don't know the English word, for the same money, you'd rather include much more patients than to have a deep sequencing. That's one of the key message. I see there are questions. Clap.
So maybe take the one from Camille Trottier. How the read length impact your results? Did you check something like this?
We were so basically we kept to only one read length. So it may be a problem for instance, it's just Illumina short reads for now, and but even in Illumina short read it can vary, and this may impact because in our classifier we used for the filtering, as it takes both into account the read counts and the fraction covered. If you have longer reads, the kind of ratio between that would change regarding to the read length, but we kept at homogeneous read length so far.
Okay, thank you very much. I just let you, Benoit, answer the question from Denis in the chat, because you have to move on with a tight schedule. And we move on to the talk from Adel Bazin. Adam is from the LabGem. He will present the result about the pangenome analysis to discover genomic islands. This is the highlights of the paper that was published in Bioinformatics last year. And Adam, the floor is yours.
Thank you for the introduction. Do you see my presentation?
Yes, perfect.
Okay, awesome. So hello, I'm Adam Bazin. I'll be talking about PanRGP today. So it's a pangenome-based method to predict genomic islands and explore their diversity. Nowadays, when you conduct prokaryotic genomic studies, you tend to rely on the comparison of hundreds to thousands of genomes of the same species when you're studying a species. Those genomes display a very high diversity in terms of gene content. Most of this diversity comes from horizontal gene transfer. And between 5 to 40% of genes between two different genomes of the same species are different. So this very high number of data and of differences gave rise to the concept of pangenome. A pangenome is a union of sequence entities shared by genomes of interest, usually from a given species, but it can be also from a genus or a phylum. It also gave rise to new data models, moving away from a reference genome that only has specificities of its own, and towards reference pangenomes, which encompasses the overall genomic diversity of the group that you're studying. So you can build the pangenome at different levels. So you can build a pangenome using sequences only if you are interested in a variation between different genomes, for example. Or you can also build a pangenome by looking at the gene content, which is what we do. So our goal is to study the diversity of genes shared by a group of individuals, usually a species. In this example, you have 4 genomes. The rectangles represent genes. Genes have the same color in different genomes if they are homologous. When you conduct a pangenomic study, you can partition the pangenome, which means that you can classify the different gene families into core or accessory genome. A core genome will include all genes that are present in all individuals of the group that you're studying. So here's red genes, for example, are present in all genomes. So they are core. The accessory genome will include genes that are present in not all genomes. So it can include rare genes or genes at intermediate frequencies. So this was the partitioning scheme that was used originally in the first pangenomic studies. However, as you can notice, it's not very stringent, as if you have gaps in your assemblies, for example, you might be missing genes in your assemblies, but the genes are actually present in the genome that you're studying. So it will drop the core genome artificially. So for this reason and some others, we use another partitioning scheme that was introduced in the Kunin and Wolf 2008 paper. The partitioning scheme goes as follows. We have the persistent genome, which is sort of a relaxed core, which will include genes that are present in all or most genomes of the pangenome. So it will include the gray gene, which was not part of the core genome earlier. The shell genome will include genes at intermediate frequencies. So present in this example, it will be present in 2 genomes, for example. And the cloud genome, which will include rare genes. So to realize this partitioning, we developed a tool called Pangolin. This tool relies on a pangenome graph. So how we build this pangenome graph, we use annotated genomes. We cluster all the genes together to build gene families. Then we build a graph where nodes are gene families and edges between the nodes are adjacencies along chromosomes. So here, the blue and green nodes are close together in the graph because they are linked in the genome. Then we partition the graph into the 3 partitions. So most other approaches rely on hard-coded thresholds that are usually picked because most people use the same thresholds. Here, we chose to use a statistical partitioning method called the neighborhood expectation maximization. It relies on the Bernoulli mixture model combined with the Markov random field to produce a partition pangenome graph. And so here is a representation of an actual pangenome graph. So this one was built from 3,000 genomes of Acinetobacter baumannii. What I can illustrate with this representation is that gene repertoire changes are concentrated in some hotspot locations and usually driven by horizontal gene transfer. In this zoom that we have here, you can see that there are 2 paths in orange for the persistent genes. And from those 2 paths are multiple cloud or shell paths that branch out. So those changes concentrate in genomic islands. And genomic islands are specific areas of genomes, of microbial genomes. They are often associated to RNA genes to integrate to insertion sequences. And they have been of very high interest to microbiologists for the past 20 years because they aggregate many strain-specific adaptations, such as environmental adaptations, pathogenicity islands, antibiotic resistance mechanisms, and from time to time, secondary metabolites as well. Those genomic islands display a very high turnover in terms of gene content, as I illustrated on the previous figure. And they also have a composition bias, meaning that they have a different GC content compared to the rest of the genome, sometimes, but not always. Because of the reason, we expect genomic islands to consist mostly in shell or cloud genes. So we chose to design a method based on the pangenome partitions to try to find those genomic islands. The method goes as follows. So we use the Pangolin pipeline that I introduced earlier. Then we project the partitions on the genes. So we annotate each gene based on the partition of its gene families. Then we compute a score along each of the genomes that we are interested in and we want to predict genomic islands in. The idea behind the score, which I will not detail here, is that stretches of shell and cloud genes will have a high score, and stretches of persistent genes will penalize this score. So in the end, what we expect to be genomic islands will be the regions with the highest score in the genomes. We benchmarked this approach against published datasets. datasets that were published originally in this 2008 paper and updated recently in this 2018 paper. The first dataset is a comparative dataset. It was generated from comparative genomics, and it includes 81 genomes from 36 different species. What we did with PanRGP is that we built 36 pangenomes using all available genomes of, in RefSec for all the species of interest. Then we predicted the genomic islands with PanRGP and then computed the benchmark. We compared the scores of PanRGP with many tools that are available in the literature to predict genomic islands, and we noticed that PanRGP came on top on with most metrics. So we were very happy about that. The second dataset is the literature dataset. It includes genomes with published descriptions and published bioanalysis of genomic islands. It's much smaller. It includes 6 genomes among 6 different species. So we built 6 pangenomes using all available RefSec genomes again from those 6 species. And once again, PanRGP came on top with most metrics. So thanks to this benchmark, we can say that PanRGP detects genomic islands very well. However, RGPs, regions of genomic plasticity, are not just genomic islands. They can also correspond to both gains or losses of genomic region. So if some regions are lost differentially in your genomes of interest, those regions that are kept in a few genomes of interest will be predicted as RGPs. They will also include plasmid sequences. So this is because plasmids are rarely kept in all the genomes of a species. So you will predict them as RGPs. And depending on the definition that you use of a genomic island, some may be persistent within a species and thus not identified as a region of genomic plasticity. I will illustrate that now with this Salmonella enterica genome. So in this representation, you have the entire genome, which is drawn. The different lines correspond to predictions of some of the tools that are used to detect genomic islands. In red, you have PanRGP. In black, you have the literature reference genomic islands. And I will zoom on one region in particular. So this is a pathogenicity island. It was described as such in the papers that described this genome. And it's of interest because it carries multiple virulence determinants for adhesion and entry into cells. And as you may notice, absolutely no tool, not PanRGP and not any of the other tools, predicts this area as a genomic island. This is because it's defined as a genomic island because of the functions that it carries. However, it does not display any GC content variation, and it does not have any integrase genes. And all genomes of Salmonella enterica that we use to build the pangenome, most of them at least, include those genes. So for this reason, it's not predicted by any other tool. So now that we saw that we were capable of predicting genomic islands, we might be interested in studying genomic islands that are found in the same persistent context. So those regions are called spots of insertion. As I said earlier, most gene repertoire changes are gathered into few hotspots. And in this example, which is a very well studied hotspot in E. coli, you see rectangles, which are groups of genes. And you see that in different genomes, you can have many different groups of genes, even though it's the exact same place in all the different genomes. So we define spots of insertion as persistent, as genomic islands that are flanked by the same genes, even if they don't have the same gene content. So since we could have so many genomes as we're building pangenomes, and since we could detect genomic islands so well, we decided to try to detect spots of insertion using pangenomes. So the method goes as follows. What we do is we take all the genomic islands that we predicted with our RGP. Then we look at the flanking genes that are persistent to the genomic islands. We compare the flanking genes together as ordered sets. If the ordered sets are similar or if they're partially overlap, we consider that there is a match. And we will build a graph where each node is an RGP, and we'll add an edge in the graph if both of the borders match. We do that for all RGPs that we predicted in the parent genome. If only one of the border and not the other match, we will not add edges. In the end, the spots of insertion that are predicted will be connected components in the graph. So we do not have benchmarks to test our method on a large scale. So what we did is that we looked for the LEX hotspots that I illustrated earlier in a metagenome assembled dataset. So we built a pangenome using 1,400 genomes from the Pazzoli et al. Dataset. Then we looked for the hotspots by looking for the persistent genes, which we found. This representation, so in this representation, each node is a gene family, and you can see colors depending on the partition where the gene family is assigned. This hotspot is found in about 400 of the 1,400 genomes. So we are very happy to be able to find it and to see that our method was capable of reconstructing the spots. So as a take-home message and in summary, I presented today methods of comparative genomics to detect genomic islands. We use pangenome partitions as a proxy for comparison. This way, the computational load increases linearly with the number of genomes, so we can work on tens of thousands of genomes. We can study genomic islands found in the same persistent context by clustering them into spots of insertion. That way we can study how the region evolves throughout a species. And all of those methods come along with a software suite, which makes subsequent analysis easier. We can output many different file formats, which can be used with other softwares. This is available on GitHub. It's installable through Bioconda in the Pangolin software suite. It's also available in the microscope platform if you have genomes in this platform. Thank you to all the people that participated in this project. And thank you for listening. If you have any questions, I'll happily take them.
Thank you very much, Adam. It was very interesting. So yeah, same. Waiting for a question. I will shoot first. You are presenting a method that is working very well to me, and but it's based on the score, and you didn't have the time to explain a bit the score. And I was wondering, so the score is obviously the score is okay because it's working. But
yes.
I was wondering if you guys check the sensitivity of the score and how much you gain or you lost if you move it a bit, and maybe how you could I don't know if you have other thoughts, especially to discover maybe missing genomic islands that were passing through the net. And so I was wondering if it was the point where you could maybe improve the method, or if you say no, it's okay, we cannot do better, and this is another game that we have to play.
So it's most likely possible to do better. We did not. I mean, we tested a few parameters, but we did not extensively try to adapt the parameters to the benchmark, because that would be cheating pretty much. But it's probably possible to do better. The scoring as it is done worked very well, so we chose to keep it as it is. But depending on the diversity of your data set, it is very possible that having different parameters can improve or decrease the sensitivity of the method. Yes. It's very possible. It's also possible that having different approaches can be better. I don't know.
my point was not to change what you did already. Okay, just for the perspective and to move on. There is a question from Leonard in the chat. It's a question about the Pangenov graph. Are they always made of unique connected components, or can they be split into multiple subgraphs if the reference genomes are used are fragmented in multiple contexts?
Okay, so that's So so the Pangenov graphs can be made of multiple components, but usually there is a single big component which corresponds to the genome. And sometimes you can have small components which correspond to plasmids. So I don't have a good example in this figure, but in other pangenomes we saw this. We saw this in other pangenomes. For the other question, usually, so if we use fragmented dataset like MAGs, we still find a unique connected components usually with maybe a bit more smaller components which can correspond to plasmids or to contamination, for example. But usually we have a single big component which is the main genome.
Okay. And there is we take the last question from Veronique. Did you characterize genes in different classes, persistent shell and low, and for example, housekeeping genes? Where did you find them?
Okay. And, so indeed, we tried to look at functions that we could find in the different partitions, and indeed, we expect housekeeping genes to be found in the persistent genome. There were some classes that were enriched in the shell or cloud partitions. I remember now that genes that are typically related to antibiotic resistance are found mostly in the cloud genome or sometimes in the shell genome, but very rarely in the persistent genome, for example.
Okay, thank you. Thank you very much for all the three speakers. That was a very interesting session. At least to me, it was very interesting.